Good evening, folks. Welcome to another NOYA Center for Marine Science Talk. This evening, our guest is Ashley Kidd, who has been up and down the coast and a really important part of this big conversation that we've all gotten quite familiar with that plays out in our own backyard and up and down the coast. And so NOYO Center folks, we hope that you will please donate. We don't really charge for these talks, but if you can donate to the organization so that we can keep bringing these fantastic science speakers to you, that would be great. Just let us know you're out there and how much you appreciate these science talks. We've had, such success with it that we might take it for granted every once in a while. But once again, just show us that you love these talks, show us that you're here, that you support science, that you love science. Science needs a little extra love and attention right now, as we all know, as do the scientists. Hug a scientist. With that, I'm going to let you go ahead and share your screen, and I'm just full of gratitude for your being here.
Well, thank you so much, and I am so happy to be here as well. Hold on. There we go. Whoop. All right. Well, thank you so much. I'm happy to present for the second time over 2 years ago. I gave an initial NOYO update, and happy to be here again. Love the Picno image from the NOYO Center. I'm the conservation program lead of the Sunflower Star Laboratory and one of the co-founders that was fortunate enough to be there from the beginning in 2021. We're a Monterey-based nonprofit committed to research and developing reliable and scalable aquaculture methods for the sunflower sea star, enabling conservation and their reintroduction. So it looks like a lot of things. The cool thing about working with a lab dedicated to a single species, it's not redundant or innocuous, day-to-day, same old. We basically did play and develop tools in all realms to help this animal's monitoring, survival, under human care, wild recovery, and develop restoration methods. So it's been really exciting. I came from working in public aquariums where I worked with thousands of different species, and that was super rewarding. But to switch to just focusing on one species has been very, very rewarding itself, being able to really delve into many different avenues of science. Here's just a little teaser of with some pictures of some of the activities that we've gotten up to in the last year with some field work, raising stars, doing some environmental DNA lab monitoring, measuring our stars, growing algae to culture larvae, and hosting our community events. So I said back in, I gave my first presentation along with Andrew Kim from the Sunflower Star Laboratory, who at the time was still Moss Landing Marine Lab staff member, but a Sunflower Star Lab board member, as was I, along with Lauren Schiebelhut, who was a geneticist who was collecting all the genetic material that she could get her hands on of sunflower sea stars up and down the coast and with the stars that were under human care. So if you can check out the recording about that, that was very exciting. We just spawned the first California cohort. So we had a male and female from the Birch Aquarium, and we've continued to raise those animals through 2026, along with other trials with different non-California stars. But it's been exciting to be able to raise these stars, and we've learned so much over the last 2 years, just raising this one family of siblings across multiple institutions. And we've also been able to do just so much work having these animals available to us in the state. So a couple highlights that we've done since 2024. We got a grant and collaborated with NOAA and a bunch of other eDNA experts at Stanford University and the Hakai Institute in British Columbia to develop an assay for environmental DNA. And I'm going to talk about that in a sec. So I'll explain what all that is if you're not familiar with it, or you were just mentioning how you were working with Cal Academy. He's using the eDNA assay that we developed. So it's very cool to see the whole community come together and use this tool. That's what it was developed for. But we did the lab and field experiments to help understand what an eDNA hit is, how sensitive the tool is, and what it can tell us. And that's been really exciting. So I'll talk about that in the talk. We tested and cryopreservation protocol. So that's where you freeze down to minus 180 Celsius. You stop cellular activity and you're able to sort of suspend living biological material and then thaw it and keep it ticking along. So we've been working really closely with the San Diego Zoo Wildlife Alliance. Back in 2024, we were able to cryopreserve sperm, but now we've done sperm and larvae. So a fertilized egg, we've been able to freeze and thaw and continue its development with the pink star. So big milestone. And now we've done so with the sunflower sea stars, but again, it's a really delicate recipe that we're working on, and that's one of the main projects for our lab. And we've been able to do field testing. We had a successful field season last year that I'm going to go over, as well as our 2026 field season, which we've been prepping for, and literally just got out of the water today with the start of our first experimental day. It's real now. So excited to share that with you, a little teaser. And then continue all the monitoring up and down the coast again, hearing Noyo Center being involved again. We have a huge coastline, thousands of miles, depending on how sensitive your coastline measurements are. And we want to start to find that southern trailing edge of restoration and recovery, of natural restoration of wild populations. So it's been really exciting to have the community out looking for them, and now having this environmental DNA tool. And sea star wasting disease, also since 2024, was identified as a bacteria. So there's been a lot that's happened. We have not been sitting back just posting fun things on social media. We've actually been doing a lot of work to support all the hype. So here we are. In case you have not heard about sunflower sea stars and you truly are tuning in to learn about them, I only have 2 slides because there's so much information about why they're important, how much we lost. But in summary, these animals existed from Mexico all the way up to Alaska. They're beautiful, the largest, fastest sunflower sea star, or sunflower star, sorry, sea star, the largest sea star you're going to see scuba diving or tide pooling up and down the coast or anywhere in the world. They can be over 3 feet in diameter, and they can galumph across the rocky reef, or here pictured in a seagrass meadow, chasing down all sorts of prey. They are generalist predators and they live in all sorts of habitats. This little graph here is a pretty sobering graph. It was published in 2021 when the sunflower sea star was listed as critically endangered, and this is the percent loss of sea stars across its global range. So it exists on the Pacific coast of North America. That's its global extent, and we lost overall 95%, and these are the regional losses. So the last time anyone saw a sunflower sea star in California was 2018, and it happened to be in Monterey. They disappeared for a couple years, and now they're being seen in Mendocino, in Trinidad, and in Sonoma. So these are all very exciting things. And the reason why we care about them, of course, is the wonderful kelp forests that provide us with so many things. They help give refuge to the largest, like gray whale, to the smallest plankton, and feed all of the invertebrates, or herbivorous fish and invertebrates that live amongst the kelp forest. They provide oxygen for us to breathe. They sequester carbon, and I mean, you guys could probably tell me all the things about kelp as well. Just a big fan, as we all are, and we want to help bring balance back to our kelp forest ecosystems, because right now we've got, as you might be aware, in Mendocino, an urchin barren issue with these lovely purple urchins just who are so very hungry and gobbling up all of our kelp, and really changing what our coastline looks like. Very quick jump back to me as a little introduction of where I came from and how I ended up at the Sunflower Star Lab. Now that we've sort of seen where the sunflower stars have come from and gone, I did move to the West Coast in 2013, where I immediately became a kelp convert and fell in love with the kelp forest. But I had previously worked in Pittsburgh, working in coral restoration. And down in Southern California, I worked very closely with the White Abalone Recovery Program at the Aquarium of the Pacific, in partnership with the federally managed white abalone endangered species. And then when the sunflower sea star was listed as critically endangered, I had to apply all of that knowledge and fascination in being part of incredibly enriching conservation programs with large partnerships, that I wanted to be a part of developing one, or jumping on what had already been a decade in the process of figuring out how and when and where we can help restore the species. So 2025 was a big year. After raising our Cupid cohort, we had 18-month-old stars in June or in September 2025. You know, they'd been living their best life. In October 24, right before we last spoke, they were these cute little sunflower stars. And we thought, if we have so many, they're all full siblings, so we won't be doing full releases with these animals, because it's not very responsible to just release a bunch of brothers and sisters to go repopulate a location. So we did a bunch of caged studies, and we continue to do so today, because all of the large stars that we have at our facility are all from the same cohort. And there's just lots of stuff that we can learn just from having stars in cages. So one of the things that we did was an environmental DNA lab and fieldwork session, and then we did survivorship and growth. So I'm going to go into eDNA, since you are probably and are working and talking with collaborators that are now applying the eDNA protocols to different regions in California. So environmental DNA, as a quick rundown, is taking DNA that's sloughed off of an animal, whether it's through just cells, slime, mucus, or blood. If you're a mammal or an animal with blood, sunflower stars and sea stars don't have blood. They have coelomic fluid. They basically take the water from the ocean, filter it through their sieve, and use it basically for all sorts of things, from locomotion to digestion to breathing and everything. So they're producing lots of DNA. All animals will release DNA as they shed all of these biological material from their bodies. And you can take a water sample and see if you can find different little DNA fragments. And in order to get a very species-specific DNA fragment, you need to look at many different relatives, like different species of sea stars, and see what the unique, this really is like the blueprint, the ATCG, those sugars that build that DNA strand. There are, it's like a zip code. You got to have your unique zip code to get to figure out where you are. So it's the same thing. You can look at your DNA strand and compare them across species and find the one that will only ever be uniquely associated with a sunflower sea star. And then you can make a primer, or you make a tag essentially that goes and hunts down for that sequence, and you can look at it and quantify it using fancy equipment. And the nice thing about that is you just need to take a water sample. You don't need to go scuba diving in areas where it might be dangerous or difficult to access. You can do it any time of year. You can take it at many different depths. If you just have a bottle, you can have a sterile bottle and take it right from the surface. Or if you have an ROV and you want to sample hundreds of meters below the surface, you can just take a water sample and see what DNA is floating around there. So that's the cool thing of environmental DNA, because it can add to visual diver or intertidal monitoring surveys to help fill in the gaps of where they are or where they might be. So the first thing you want to know is how much DNA do stars give off. So we did this little study in the lab where we had 12 different sunflower sea stars. So that's one of our stars, each in a aquarium tank. And then we just took water samples for 4 days to look at how much DNA was being sloughed off into the water. And Megan Shea in the background, she's the postdoc who was doing all the post sloughing experiment with us. And then we also looked at how long the degradation happened. So on the previous slide, I had light, temperature, microbes, and time can all degrade DNA. So you kind of want to know how long DNA hangs out in the water column. So you can say, okay, if we get a DNA hit, they've definitely been here. They've been living Pycnopodia in the last 24 hours, say. So that's Megan Shea and Laura Kubiatko from Stanford, who came and worked with us at Sunflower Star Labs last summer, and MJ, our lab tech who was helping out during that time. The second part of our environmental DNA experiment was in the field. So we put 12 sunflower sea stars in these cages. They're oyster cages, a commercial oyster cages. We plopped them in the middle of that orange crosshairs. And then we took water samples, those eDNA samples, at certain times over a whole week using free divers. And this was one of the coolest applications of free diving for science that I've heard of, where we had 16 samples at minimum taken at the surface or at 30 feet below. And these are all free divers that just had their first level certification to 30 feet. And we trained them very briefly on the method, that lovely hand wavy method. You got to pretend like you're a whale shark just slurping up some water. And that's it. That's collecting a water sample with a free diver at 30 feet. And then we put those in filters, and then the fancy extraction of the DNA from just a little physical filter is what takes a little bit more practice. So we had a wonderful group of Monterey freedivers, as well as the Peruvian freediver record holder who just happened to be training in California and came and helped take our water samples. So that was really exciting. And yeah, it took 5 minutes to collect 18 or up to 34 samples. So that was very cool. And we did that for a whole week. But then the processing, so we have to drain all that water down into these little Steravex filters. So it's just a fine micron filter, and it just captures anything that's in the water so that they can extract it later. And then this is a little time lapse of Megan processing all the samples over a day, just so you can appreciate all the hard work and so many skilled hand-to-eye movements to process all the like 270 samples that we made. Then we had a lovely PhD student, Maddie Pacheco from UCLA, hear about us through an outreach event at the Santa Monica Pier Aquarium. And she came and saw my talk that I gave at the Aquarium of the Pacific last June. And she's like, I want to do pycno eDNA work. And I'm like, great, we just developed an assay. And with the help of another graduate of UCLA's Environmental DNA Lab, Zachary Gold, who now works at NOAA's Pacific Marine Environmental Lab in Washington, we wrote up a little grant and got funding to do a blitz of the entire coast. So this is us, or this is a Maddie in the back with the orange Grundon fishermen set up there. We're staging all those environmental eDNA bags. And this is Dr. Sarah Gravum in our lovely Sunflower Sea Star hat, who helped finagle this luxurious yacht to go from San Francisco all the way down to Santa Barbara to do a big stretch of the coastline. And this particular picture is very important because this is actually at Half Moon Bay at Mavericks, and it just so happened to be the furthest south hit that we got at Mavericks. So this is a little map of the positive and negative hits that Maddie got from going up as far as Mendo and all the way down. We technically went all the way down to Santa Barbara, but the map cuts off just in Santa Cruz. But this is a great win. This Half Moon Bay would be the furthest south that we've got any sort of positive validation, whether eDNA or visual. And that could mean a lot of things, whether it's planktonic larvae or adults that are hanging out at Mavericks. Again, that's a huge surf site. It's really hard to dive. It's not really well surveyed. So there could be pycnos hanging out there. You don't know. So this is very exciting. Just from 1 year's worth of environmental DNA data with an assay that was only developed like 13 months ago as of today. So it's pretty exciting. And then we also were able to do a crazy cool environmental DNA. I was telling you how you can take water samples even at depth. This is at 300 feet on rebreathers with the NOAA technical dive team. Doing the same hand wavy whale shark collection. That was taken by Dave Whitting, who's the NOAA habitat restoration lead, and just an incredible collaboration between the Channel Islands National Marine Sanctuary staff, boat ops, the NOAA Technical Dive Team, Sunflower Star Lab, and California Department of Fish and Wildlife. So that was another incredible eating. You could not have, this was not planned this time last year. So that's the EDNA stuff. So we can chat about that in the discussion after if you'd like. It's been a wild time, but the other thing that we did was just put stars that were raised under human care at the Sunflower Star Lab in California Academy of Sciences. For me, it's up in San Francisco. For you guys, it's down in San Francisco. In Monterey last year, so immediately preceding the environmental DNA free diver experiment, we went straight into a month-long survivorship and growth. So basically putting out stars that have never touched the ocean to go out and spend a month. So we called it Camp Ocean. And this is Vince Christian, the true founder of the lab, the brainchild of the Sunflower Star Laboratory, carrying the first set of sunflower stars down to the depths. And then this is actually True Barnes, one of the Reef Check Central Coast managers, who's one of the partners helping set up the site. And so we had, again, 4 sunflower stars in these cages. We had paired cages across 6 different derelict or deserted mooring sites just off of Del Monte Beach in Monterey. And we had 1 cage with 4 stars from Cal Academy and 1 cage with 4 stars from the Sunflower Star Lab. And all we did was feed them, check. We developed health metrics. We took instead of a blood sample, we took coelomic fluid samples before and after to see if their microbiome changed or if their health changed or deteriorated, if it had any, if it actually showed up in the internal coelomic fluid changes. But the good news was all the stars did really well. They all survived. Wasting, but we did have 1 cannibalism event because you can see, and I know you appreciate some rough ocean conditions, but last week we were pushing our field season pretty late. We had the stars in through the end of October, and there was a really low vis event, and we literally could not find the cages. So we skipped a feed on one of the sets of stars. And when we came back the very next scheduled time, one of them had been eaten by their siblings. So fun fact, sunflower stars are cannibalistic. And yes, they just love to eat one if they can crawl on top of it. Not all the time, they are also gregarious. So it's a fun dynamic. You're like, are you guys hugging each other or are you guys gonna eat each other? And this is just a beautiful, beautiful shot by Ralph Pace, who captured the event, the transfer of those delicate little stars. So these were about 10 centimeters last year. So this was in September of 2025. And they were about 50 grams because the 12 stars was 600 grams. So 50 grams. So just think of like how much your cup of coffee weighs in the morning. It's not quite that. We went out and fed them frozen kelp. So this is Ellen Umida from the Monterey Bay Aquarium who came out as well. One of our other field partners that dedicated boat time and divers to help us do all of this work. You can tell there's a little bit of surge. So feeding frozen krill in a surgy environment requires some timing. But that was fun. And that was a little snapshot of our partners from last field season. And then this field season, this was just from last week. We literally just set up the site. We're putting more cages and doing a different set of experiments now, just further down the way at Tankers Reef. And we had a great group of divers. This photo of the group of us was literally taken just a few hours ago, after we came out of the water. So it's been an incredible year. Lots of work. I could go off on so many tangents, but I'm going to hand over the questions to the room and see where we're at.
Excellent. Thank you for all of your incredible work, Ashley. This is so exciting. I hope that people will chime in. If you'd like to, let's see, where's my chat box? I have your screen. Maybe if you stop share, there we go.
Oh, sorry, you could see my, I see. Yeah.
That's so good. Now I can, if folks want to put questions in the chat, you're welcome to do that. Or if you prefer, just to raise a hand, and I think we can spot you. And don't be shy. If you have a question that crossed your mind, hopefully you can remember and ask away. Otherwise, I will. Or I'm just scrolling across folks here.
Or I might just start asking people questions. Or hands up, how many people have seen any of the returning Sunflower Sea Stars to the North Coast? Anybody on the call? Yes, Abby.
Abby. Nice. Yeah, there we go. Well, Abby's seen some, that's for sure. And if we look real locally, we had this superstar, as you know. Heather's seen one. I think we saw that the superstar in the cave here in Mendocino that we grabbed a little DNA from when you were up and looking with the whole crew. I'm kind of curious what that showed, if that individual was actually related to who, or if you were able to get any.
Yeah, so it's funny because we did take that sample in July of 2024. And again, that is what instigated that first talk. Yes. First talk in November 2024. And Lauren Schiebelhut, who's the geneticist most directly involved with the Sunflower Sea Star genomics work, is literally assembling, because we had the Mendo star, and then last summer we also had those Sonoma stars that were seen, and the genetic samples were taken from them as well. And there are more stars that Cal Poly Humboldt folks have helped collect genetic samples. So that was all last summer. And so she's literally, again, because she teaches at a community college, so she can only do the research in the summer because she's so busy making sure her students are learning all the cool things during the school year. But she's assembling all that collective genomics work of new sunflower sea star sightings in California right this very minute.
Perfect.
So yeah, and you're like, some science works really fast. Like the eDNA assay was wild that we had a grant we applied for, we got funding. In April of last year, we didn't have an assay, but in May we did, and then that just set off a huge domino effect where all these eDNA opportunities were not even planned out. It was just running with it. And then the genetics, when you're trying to like source all these, but running the genetics, you want to have like a good sample. So you're waiting for all these samples to come in and permits to go through. And so, yeah, it's wild to have the two sides, it's never fast enough.
Yeah.
But yeah, hopefully we'll be able to share the genetics of your how is your local sea star doing?
Funny you should ask. I don't think it's been spotted in, I think I saw it in November 2025 was the
Mhm.
last time I saw it. And then so it's either moved away, moved out of that immediate area,
Yep.
or some, I mean, there were, it got real famous and there were school groups and people coming. I mean, I was kind of grateful for the slippery, muddy, eroding trail at
Right.
times, and then the limited times that people could actually get over there. But boy, when it was low tide, we had a lot of visitors. That animal had a lot of visitors.
Yeah.
So fingers crossed, it ran out further out where there were more urchins or something, because it was hungry. There was, times when there were a little handful of urchins sprinkled,
Yeah.
and it would just grab them and eat them. So I don't know when the most recent sighting has been, is my point. Um,
Yep.
so, and then also just with the genetics, take all the time it takes. These things, this good work takes a while, and we can be patient. Um, I do see some questions popping up in the chat. And this is from Mark. Brokering, are you planning to introduce genetic diversity into the stars in your lab? Great
Yes, this
question.
actually perfectly aligns with the whole genetic discussion. Again, you don't need genetics to make decisions on obviously having more than one male and female as the parental cross is ideal for having and what's really cool and important to remember is there are very few in California, but there are still millions of sunflower seed stars. They're just way, way, way far away. Um, and even though these animals, I didn't talk about their reproductive cycle or their life history, but they do spend over a month um, just floating as little baby larval bodies in the plankton and can travel, in theory, if they're not eaten, as they are plankton and lots of planktivores will eat anything that's in the plankton. Um, they can travel for hundreds of miles, if not thousands, in theory, as long as there's the right conditions, and they can settle very far away from their parents. But the question is California willing and patient enough to wait for natural recruitment, and how is that going to affect the kelp forest ecosystem as a whole? So to answer Mark's question, are you planning to introduce genetic diversity? Yes. Now there's two things. A, we need just stars that we can pull from. Um, and then there's the permitting um, issue of not being able to, or it can take a while to bring the stars in, and then the justification of like why these stars versus local stars. And then there's the other question of, well, we see stars in the ocean right now. What does the community in all these different areas feel about uh, taking genetic material or broodstock out of the ocean in order to increase the genetic diversity in the stars in our laboratory? So these are questions and discussions that we are hoping to have or planning to have this summer with our well, with people that would be working, well, that we've been working with, that we would want to have their input in on what are the best ways forward. There are many different options that have been applied to corals and abalone and other marine invertebrates, and it's really understanding why, when, and how if we were to do that. So we are there. The fact that we've started to see back in 2024, we just had the beautiful Mendo Pygno, really. And now 2 years later, there have been, well, there were 19 stars. There was a National Geographic article that just came out last week, highlighting all the work that happened in Sonoma last year. I don't know if you've come across it or has anyone read that article that came out, but you can search for it. Or I can make a note to send it in the link, but there were 20 sunflower sea stars found just in one cove in Sonoma. So we know that there are, there a burgeoning, but still very small population of picnos uh, in the North Coast. And oh, thank you, Abby, for the link to the National Geographic article. And we are working on it. We're just trying to make the most of what we got and making sure that our partners that are working in restoration are in the conversation. So thank you for that question.
Absolutely. Yeah. We were the divers, the Kashaya divers, I think that was the site that we've been kind of partnering up with, and divers went down and saw them. I don't know if it was that same group or if there was
Yep.
yet another sighting of more recently. I'm not sure. Okay.
With that group. Yeah.
That group, super exciting. I remember the day that Richard Millis came back to staff meeting and had that to share, and it was like, what? So exciting. And then do we know more about the bacteria that affected them, and if it's still present, and or has it mutated at all, that you know
So
of? Corinne asked.
thank you, Corinne. That's a great question. And what I my response when they were asking um the Sunflower Star Lab, the Sunflower Star Laboratory, once the publication came out identifying the bacteria as Vibrio pectinicida, they're like, oh, well, what are you going to do? And I'm like, well, it doesn't really give us any answers. It just gives us at least something to now be able to look for. And so it's really it was a phenomenal effort to identify. I mean, it took them years to uh, develop and conduct the experiments to really hone in on that particular bacteria and doing all the different tests that showed that no, truly there was causation or and not just an affiliation of a bacteria that would just be colonizing when it's if a star is sick. But there are so many things we don't know. So we don't know if it's been mutated because we don't have a sample of what the bacteria was in the stars during the time that they were wasting, or in Hakai they may have, but I don't know if it is unclear to me whether they've been able to look at whether it's mutated. The other thing about that is maybe there are regional differences in the Vibrio. We don't know. There's only been confirmed Vibrio from these experiments in British Columbia and Washington. And so what we're doing, we also in our permit in collaboration with California Department of Fish and Wildlife, we are collecting coelomic fluid, so the not blood, but the liquid within the sea stars from areas in Monterey Bay, because that's our backyard. And we are starting to take samples from stars because it wasn't Vibrio pectinicida can be found in all sorts of some sea stars. And we're trying to see if we see the same bacteria down here. Does it look a little different down here? Is it the same? If we see stars wasting today, do they have Vibrio pectinicida? And then just to go back to the Picnopalooza, as it's called, where the Kashaya and San Jose State and 25 other scientific AUS divers went to Sonoma to sample all these stars last summer. It was right when we were developing, that's like we had an eDNA assay, and so we took a water sample right above a sick or wasting sunflower sea star in Sonoma. And we know that when stars are wasting, they produce a lot more eDNA. And of course, the DNA of the bacteria is also in the water as well. So we're hoping to be able to get that processed and see. That would be like the first time California wasting star would have had would have had a test for whether Vibrio pectinacida was in the water or not, like a meter above the starfish. So yeah,
Oh,
there's a lot. There's just so many places that we can look and investigate to try to figure out how and when and what levels this bacteria uh exists and what that means to sea stars in general and specifically to sunflower sea stars. So yes, it's again, we're just back to square one again and just asking all the questions now that we have something to look for. But it's been really wonderful to be able to work with the Department of Fish and Wildlife to support um them. They're processing all the samples that we're taking for them uh and developing protocols. Again, that's all in collaboration with the Hakai Institute that um, the primary author of that um, Vibrio pectinacida paper works at. And so we're working with the people right at the source to sort of make sure that what we're doing in the field to help monitor for this bacteria is truly helping us understand what's going on.
right. It's so fast paced and so sort of condensed and so like not knowing what happened and then it happened and then figure that out and find this. And it just seems like such a fast paced sort of experimental phase of this. And now
Yeah.
I wonder, oh, well, Sharon is wondering if they prefer a particular water temperature, which sort of leads into what I was thinking as well around. I'm sure you've heard it 150,000 times about the upcoming more warming period that we're all kind of
Yep.
looking at coming our way, since that was sort of the impetus of what happened last time. But yeah, Sharon just yeah, preferring water temps. So as they're floating around as gametes or whatever, do they have a preference? I mean, they're all up and down the coast.
Yep. And generally, surface sea temperature is warmer than average sea temperature. And um, to answer the question, I can answer this one straightforward. They are generalists. They tolerate like anything from 4 degrees Celsius to 18 degrees Celsius in the extreme. So they're pretty forgiving in terms of what water temperature they live in. Um, what's interesting is, do they know that staying in warm temperature is going to allow infections to progress more quickly in them? A really interesting observation from researchers up in British Columbia is that the sea stars, yes, we've even seen them like in Southern California and Anacapa. They would live year round in, warm, like 16, 18-degree water, like, you know, 10 feet, 20 feet. They were just hanging out. But in British Columbia, much colder, but they would also have warm sea surface temperatures in the fjord. So like the like little deep finger projections from the ocean going inland. And when the snow melt from the glaciers in the fjords would create a lens over the sea, it would force the sea stars lower because what they are not tolerant of is low salinity. And actually, they think that's what might have spared them because it would force the stars into deeper, cooler water in this that stratified those layers in the fjords, and they didn't waste compared to their outer island sea stars that didn't have that lens and they were just hanging out in warmer surface temperatures, and they are seeing a failure to persist into those larger sizes. So they would settle out as stars, but they haven't seen like stars continue to grow. They think that they actually are wasting during spikes. And we do see or not me personally, because I don't do intertidal monitoring along the coast, but with all of our wonderful partners that do all of these things, they note that typically you do see wasting spikes after the warmest months of the year in September, October, once the seas are warmer. But it's not that the stars are sensitive to the temperature. It's most likely the fact that just biological processes speed up in warm temperatures, and the bacteria can just grow and multiply faster and spread more vigorously in those temperatures. But
Nice.
isolated wise, they are fine in warm temp and not fine. I mean, you don't want to like roast them and have them in like 20-degree water, but actually last summer when we did our field experiments, it was in September, October, and there was a heat wave when we were doing our environmental DNA field surveys or field experiment, and they looked great. Like we went and saw them every day. Our divers went down and they looked amazing. So I just from an anecdotal observation, again, these stars was the first time they saw the ocean. It was the first time they were in that warm water, and there was no bacteria present. And again, this has been reiterated by also the folks at Hakai in British Columbia that, yeah, in the absence of a pathogen, they can tolerate warmer temperatures just fine. It's that pair.
Wow. Beautifully answered. That's really, really kind of puts all those moving parts in a concise understanding. So, does anyone else want to have a little question? Don't be shy. I saw Heather King. When was the most recent sighting that you had of a sunflower sea star? I know you're down all over the place in the intertidal. Hopefully you hear me. Don't be shy, Heather. Pop off. She's being shy. Okay, she said fall 2025, and she can't remember the exact date.
Well,
So
to
yeah,
give you hope, there have been sightings more recently than that. And iNaturalist truly is a wonderful treasure trove of Pycnopodia sightings. And they have been sighted, I mean, definitely in Humboldt. And I haven't looked recently for Mendocino, but I know that in this year we've had sightings of Pycno. So they are not gone. However, to your point of, did you ever, I'm just curious, did you ever name or did people come up with names for the popular star?
yes,
What
yes.
were they?
Gollum.
Yes, that was the one. Gollum is the name I also gravitate to. He's so Gollumy, or she.
He is. Yeah, he or she, definitely. Really hung out there near that yellow sponge, which looked like it had some chomps out of it. I mean, when you were here, they were in the middle pool and then spent like many, many months kind of over on the side of the cave, which was interesting. And what's really interesting, one of our local tide poolers was one day I thought saw a different one, and then that one I don't know was through the little cave and on the other side. But
What?
anyway, yeah, I know. So yeah, iNaturalist is great, and we just had a BioBlitz at Van Dam, like I
Oh,
said,
yes.
and just got into the water, and that crew from Cal Academy was actually taking water samples and showing us how to do it. And the NOYO team was excited to kind of learn how to do that. And also just really hoping that, we're always ready to get out and do help support and do iNaturalist and just be out there and contribute in our community science way that we can. I've got this whole slew of beach surveyors who pick up trash, report deceased marine mammals to me. And then they also count bull kelp, beach cast bull kelp, which they've been doing for 5 years. And we have all of those data on our local beaches. and we're kind of going, well, why are we doing this? Why are we counting kelp? Well, it's just to see sort of seasonally, and then they're also noting reproductive spores. Um, but they're like, okay, we'd like to do something more than just count kelp and pick up trash and report marine mammals. So, so we're just kind of giving that some thought. And then being, collaborative with the INAT community in doing just getting out there and collecting more data and paying attention, right? I think that's really what I'm trying to say is that these different situations kind of snuck up and bit us in the butt before we knew what was happening. And now this time people's heads are wrapped around it. And so they're more like, oh, what does this mean? Is this going to cause this and this and this? And so people are just way more aware and wondering, how to keep track and how to monitor and how to keep listening to what you're seeing and what you're learning and stay in tune with what the scientific community is talking about. And those conversations just seem so gosh darn important. And I hope
Yeah.
you're getting support. That's the other piece of this scientists in this current atmosphere. It's hard, right? So I'm really hopeful that you continue and all the people that are doing this work continue to feel the support. If not all the love, but also monetarily, it's got to be, you guys are incredible what you're doing, all the work underwater and
Yeah, I mean, I will never shy away from that. Our our donation button on our website is active, and
great.
if you have any collaborative ideas of to help bolster our our funding to keep doing what we're doing, reach out to me and I'll put you in touch with we have a development committee.
Yay.
So again, yeah, that's we need the funding to keep doing what we're doing, and again, the community support, the turnout to do this work requires so many people, and always so thankful for people showing up.
Right.
One last thing before, I know it's past 7, but how can we check in with the BioBlitz results was a question, Heather. I know Cal Academy does a report at the end of the year. They do have a page on their website on their BioBlitz. I don't know how, like, from a like signing up for a newsletter to make sure you get it. I'm not aware of, but I can check in because I talked to um the folks at Cal Academy all the time on this because I mean, it is the work that we're doing together that's enabling us to start rolling out, environmental DNA stuff into their BioBlitz, and thinking of community engagement. How do we do eDNA sustainably over the years up and down the coast? So
Right.
those are definitely things that require funding. So if you like eDNA and you want it, we can start building up that capacity.
Cool, cool.
Oh, thanks,
Yeah, I
Mark.
see that.
There you go.
Oops. Oh, great.
Cal
Perfect.
Academy's website, a whole thing on BioBlitzes. Yeah, perfect. Thank you.
Little clue is to not just try to do it on your phone, but to actually go onto your laptop or computer. And that platform is a little bit easier to navigate in terms of specifics. And then I just saw Abby's comment that she saw a sea star a few months ago at Noyo. Woohoo! So
Oh,
great.
yes, Abby. Sorry, of course. It was actually because of the eDNA hits at Noyo, like at the beach. It was on the Reef Trek transect, like on their survey line. There was a little baby picno that was seen.
Oh, that is
Yes,
so cool.
yes,
She said not me, but the Reef Check team. That's that
yeah,
I forgot. Yeah, super cool.
yeah,
Fantastic. And then Rebecca and et al. That from Cal Academy, they went and did a survey today with the Kashaya at Moat Creek. That was where the next BioBlitz was down there, and they were also sampling water. So
yeah.
um, yeah, if you don't do INAT stuff, get on there, do it yourself, or peruse what people are seeing. It's just really, really cool stuff. So, well, we will let you go get warm and maybe get the salt off if you haven't already.
I
You
did
probably
shower.
just
I did.
Good. All right. Well, everybody, thanks for being here, and keep showing the support, keep showing up, and stay curious.
Yes,
Yeah.
and thank you again for inviting the Sunflower Star Laboratory back.
Ah,
Yeah.
always, always. Yeah. Thank you so, so much. It was great to see you.
Yes, and thank
Look
you for all your
forward.
questions.
Yeah. Great questions, folks. This was really a lot of fun. So enjoy, and we will see you all next time.
All right, take care. Bye.
You too. Thank you. Bye bye.